Hi students, this is a description of mitosis and meiosis and the consequences of those types of cell replication. So we have on the screen mitosis. This is the way in which most of your autosomes or your body cells replicate. So we have here a model of a cell. It could be a skin cell, it could be a kidney cell, it could be a heart cell, it could be any cell except for your gametes, except meaning for your sperm and your egg. So we're going to keep this pretty simple. There's going to be three stages essentially for mitosis. So in this first stage, we have this DNA replication phase, where inside of the nucleus, the chromosomes have to make an additional copy of themselves. So in humans, most humans have 46 chromosomes. In this phase, we will see 92 chromosomes inside of the nucleus of the cell that is replicating. Then we have this second stage that I called the DNA segregation phase or separation phase. The idea is that the chromosomes, all 92 chromosomes, line up in the middle of the cell and then are pulled apart so that 46 go to one side of the nucleus and 46 go to the other side. And then we have the third stage, which is the cell division, where each nucleus divides in half, and each nucleus and then each subsequent cell has 46 total chromosomes. So as you can see, this is a cycle. This cell will at some point grow and will replicate this phase again on its own. Likewise, this cell will inevitably replicate and undergo this process. So all of your cells will do this over and over again at different speeds. Your skin cells, your liver cells, your stomach cells, the cells of your large intestine, those replicate very quickly and frequently because they tend to get a lot of wear and tear. Your stomach acid eats away at your stomach, so you need to constantly replenish those stomach lining cells. Your skin is constantly damaged, injured, bombarded with solar radiation, so your skin is constantly the top layer of skin is constantly being removed, sloughing off, and your skin cells underneath are constantly growing and regrowing. Other cells, like your heart cells or your brain cells, your nerve cells, grow very, very slowly. This is why damage to those types of cells is very serious, because those cells do not have the same capacity to regrow as your liver and skin cells and all those other cells that I talked about. Now, your sex cells, your gametes, undergo a slightly different process that we call meiosis. So your sperm and eggs, the sperm in a man and the eggs in a woman, undergo these three stages, but they have to undergo an additional process. The reason they have to undergo an additional process is because they need to split the total number of chromosomes. Every one of your body cells, your autosomes, need to have a full set of your genes. Your sperm or your egg can only have half of your genes because they need to fuse together with the complementary gamete in order to produce a functioning human being. So we're going to keep this simple and say that after your sperm undergoes mitosis, it will undergo another round, another set, a second division in order to have half of the total chromosomes. So where we started with 46 chromosomes and it became 92 in the replication phase, and then split up into two cells of 46 chromosomes. What we see here is that these 46 have to split again so that each cell now has 23. So in a man, every single sperm that he produces will have 23 chromosomes. In a woman, every single egg she produces will have 23 chromosomes. Men produce significantly greater amounts of sperm. They produce thousands of sperm every second. Women produce, on average, one egg cell a month and release one egg cell a month from their ovary. Now, when a healthy sperm with 23 chromosomes fuses together with a healthy egg with 23 chromosomes, you will have a functioning human being that is potentially grows to be a fully functioning adult. Sometimes there will be errors in this phase of meiosis in which a sperm or an egg might have too many chromosomes, in which case the resulting fetus, the resulting embryo, might have additional copies of chromosomes. We can see that in individuals who are born with what we call trisomy 21. This condition is the result of having 47 chromosomes instead of 46 chromosomes. Individuals who are born with Down syndrome have a third copy of chromosome number 21. So what we're looking at here is a karyotype, a map, a genetic map of a boy. We know it's a boy because the sex chromosomes, the X and Y chromosomes here, indicate this is a male. And we see 3 copies of chromosome number 21. Normally we have 2 copies of chromosome 21, but individuals who have Down syndrome will have 3 copies. Now, having 3 copies means that they're making additional proteins, and those additional proteins have an effect on the development of the cells of this individual. And among those developments we see are delayed physical and intellectual capabilities. So on average, individuals with Down syndrome, their intellectual capacity tends to be limited. In certain capacities, again, this is not trying to say anything about their value as people. It's simply that they develop in a different way, and that those developmental differences are the result of having a third copy of the 21st chromosome. Other conditions can result if there are additional copies of other types of other chromosomes. There's trisomy 18, trisomy 13. Generally, those are very fatal. The individuals who are born with those will not live very long. Individuals with a third copy of their 21st chromosome can live a mostly full life. Sometimes individuals are born with additional copies of the X or Y chromosome, and those produce their own types of developmental differences. But while we can't say exactly for sure what makes it likely for someone to have an additional chromosome, one thing that research does seem to indicate is that the age of the parents has a increased probability of children developing Down syndrome or other genetic conditions. There seems to be some relationship between the age of the mother in particular, although it can also be the age of the father, but the age of the mother seems to have an impact on the probability of developing Down syndrome. So essentially what the understanding seems to be is that a woman in her mid to late 30s is more likely to have a child with Down syndrome than a woman who is having her children in her 20s.